Ja, hallo zusammen. Mein Name ist Susanne Weirich und ich bin heute hier, weil mein Partner vor über 5 Jahren infolge einer schweren EBV-Infektion an ME/CFS erkrankte. Er hatte damals, wie wohl alle anderen Betroffenen auch, sehr vieles versucht und ausprobiert, um wieder gesund zu werden. Leider alles ohne Erfolg und mit zum Teil sehr schädlichen Auswirkungen. Von Seiten der Ärzteschaft begegnete uns viel Inkompetenz, Ratlosigkeit und bisweilen auch Arroganz. Durch eine Gutachterin der deutschen Rentenversicherung wurden wir sogar kriminalisiert. Als unsere Verzweiflung infolge der Entwicklungen besonders groß war, sind wir im Internet auf Professor Ron Davis und die Open Medicine Foundation gestoßen. Bei einer Veranstaltung der OMF und der italienischen Patientenorganisation in Norditalien im Juni letzten Jahres konnte ich Linda Tannenbaum persönlich kennenlernen. Im September letzten Jahres bin ich einer Einladung zum 2. Symposium der OMF nach Stanford gefolgt und habe dort auch das Chronic Fatigue Syndrome Research Center besucht. Inzwischen sind Linda Tannenbaum und Professor Ron Davis mit der OMF zu unserem Rettungsanker geworden, ohne die wir die unhaltbaren Zustände in Deutschland nur schwer ertragen könnten. Denn während in Deutschland die Politik und die medizinische Fachwelt, von einigen Lichtgestalten abgesehen, heute noch darüber diskutiert, ob die Erde eine Scheibe ist, sind unsere Freunde in der neuen Welt und einigen anderen europäischen Ländern bereits Lichtjahre weiter. Und deshalb freue ich mich außerordentlich, euch allen heute Linda Tannenbaum vorstellen zu dürfen und den folgenden Beitrag mit dem Titel Forschungsüberblick Juni 2019 ankündigen zu können. Ich hoffe, dass dies allen Betroffenen Hoffnung darauf gibt, dass es eines Tages ein Leben nach und ohne ME/CFS geben wird.
Well, hello from Los Angeles, California, and thank you for inviting us to talk today. I'm really excited to be with you all, and this is wonderful to be able to do this from Skype. And like many of you, I'm a parent, and I started Open Medicine Foundation because ME/CFS, as you know, is misunderstood and underfunded and under-researched. And as you're aware, something must be done to help all the people suffering with this debilitating disease. So we started this foundation to do something about it. And we've prepared a video for you, as you know, with German subtitles, so to present to you our exciting research studies that we're doing. And we're planning to do this at our 3 ME/CFS collaborative research centers that you'll hear about in the video. And we have a scientific advisory board with 2 Nobel laureates and 17 top world-renowned scientists. And we work with about 100 different scientists and clinicians in open collaboration for the purpose of finding a diagnostic and a treatment, and of course, ultimately a cure for ME/CFS. And we do this all with private funding and a lot of time spent on fundraising. So I wanted you all to know that we're having a 3-day science meeting next week, and we're having a community science symposium next Saturday. So if you haven't signed up, it's free on live stream, and you can go to our website and sign up. And it's also on our Facebook. It's going to be Facebook Live. And because of the time difference, you may want to watch it later. We're videotaping it, and we'll put it up on YouTube in a couple of weeks so that you can watch it at your leisure. But there'll be about 8 or 10 different scientists giving a talk on the current status of the molecular basis of ME/CFS. And there's so much research happening now, and much more than ever, as you know. And we're very hopeful that breakthroughs will come soon. There's a lot of researchers working on it. There's a lot of people that are interested in working on it. And the more money we have, the more we do research. So we have hundreds of people now available with their expertise to work on this, unlike never before. I mean, 10 years ago, there probably were 3 or 4. So this video will give you a picture of some of the research that's been planned, and some of the research that we are funding, and some of the research that's going on at our centers. And then Chris Armstrong, our science liaison, you'll meet him in the video, and you'll also meet him at the end of the video. and he'll take your questions, any questions that you have on the science that we have presented, which is why we have him answering at the end. He's a scientist, and he can answer science questions if you have some related to some of the research projects that we're doing. So I wanted to thank you very much for listening and for your interest and for being here and for helping everybody find a cure for this as quick as possible. Thank you very, very much.
We want to thank you for your continuing support that has allowed us to do all the research that we're going to talk about today and all the research that we hope to be able to start at our new centers. So we're excited to have you here, and we've decided to do a webinar this time because we have so many items to talk about today, and we wanted to have you be able to follow along on our PowerPoint so that you can understand. So I'm also honored and thrilled to be able to introduce to you, most of you already know him, Chris Armstrong has joined us as our science liaison, and he has moved up to Stanford and is going to be working with Ron Davis up at Stanford. So he will be talking in a few moments and letting you know what studies that we're funding already and those that are on the horizon for our new centers. So I want to take you a little bit through where we're at, and then Chris will take over. So most of you probably know that we have funded the launch of 3 collaborative ME/CFS research centers. We are thrilled to be able to have done that and have really increased the number of researchers that are not only working in this space, but are talking and sharing openly the research that we're doing and that the ideas that they have and the research that they're doing. So the first one that we opened, as you know, is at Stanford, and we've been funding research projects at Stanford since about 2014. So this is all under Dr. Ron Davis. He's the director of our scientific advisory board, and he's the director of all of our collaborative centers, along with the other co-directors that we'll announce in a moment. And Dr. Ron Davis is at the Stanford Genome Technology Center, as you know, and that team has grown tremendously over time. And we'll talk about some of the great studies that are going on there. And the Harvard Center that we opened up last year and officially launched a few weeks ago, actually, the ME/CFS collaborative research activities at Mass General and Harvard-affiliated hospitals is under the co-directorship of Dr. Ron Tompkins and Dr. Wenzhang Zhao. And we're very excited to tell you of all the different activities that they will be doing there, and Chris will go over those in a few moments. So a few months ago, we launched the start of the Collaborative Research Center at Uppsala University in Sweden under Dr. Jonas Bergquist. And with all of these 3 collaborative centers focusing on different aspects of the disease, working together, really complementing and working together to find an answer for this horrible illness and to really find out what's going on molecularly, to do some clinical studies, to do some clinical trials, and really to bring this to a much higher level. We are working all together with other people who are doing research all over the world, and we're really proud of where we're at, and we encourage you to follow along. And we are taping this, so you can listen to it later if you wish as well. And if you need to get off, you can listen to it later, and we will send that out as well. So this is Ron Davis. As you all know, he's our director of our scientific advisory board, and he's also the director of the Stanford Collaborative Research Center. So the founding members of the Stanford collaboration was in 2014. It has really grown since then. So there's a lot more researchers that are part of this collaborative. But what we wanted to show you here was really how we're collaborating with other universities and other institutions. And this is a full blown out effort with experts in the field, experts in many fields. And so we wanted to give you a look of what other groups that we're working with as well. Last year and this year again, we're having our 3rd annual science symposium. It's a 3-day science meeting followed by a full-day community day. This is last year at one of our science days, our Stanford Collaborative Research Center group. And we also have the community day happening. It's going to be the week of Labor Day. We'd love for you to join us. If you can't join us in person at Stanford on the Saturday after Labor Day, we're going to be doing a live stream, a free live stream, and we'll also be videotaping it, so it'll go up on YouTube so you can listen to all the speakers. The Harvard collaborative research activities at Mass General and Harvard-affiliated hospitals is, as I said, under the directorship of Dr. Ron Tompkins and Wenzhang Zhao. And the founding members of this group, we got together a few weeks ago in Boston. We had a really great science meeting, and we have a growing number of researchers and clinicians that want to join this effort. And we're going to be doing a clinical look at ME/CFS patients at Harvard and, as you see, a lot of other universities and institutions so that we can, again, all work together. And this is a picture of the science group. Most of the members that came to the science meeting are in the picture. And this was just a few weeks ago in Boston. And then the following day, we had a half-day public seminar. And we have taped that, and we will release that within the next few weeks so that you can hear the talks that we did at that public forum. This is Dr. Jonas Bergqvist out of Uppsala University in Sweden. And we just launched that a couple months ago, and Dr. Bergqvist has already pulled together a really great number of researchers and, again, universities and institutes to really work together for this collaborative. We've actually been funding research of Dr. Bergqvist and the late Dr. Bloomberg for quite a while in Sweden, and now we are excited that we have an official center, an official collaborative research center, so that we can pull others together into the fold and really grow the work that we're doing there.
All right, thank you very much. Do you want me to jump in here, Linda?
Sure, go right ahead.
Okay, great. So this is the basic overview of the research strategy that we've come up with. So this is a slide just to encapsulate kind of all the research that is going on, just to simplify it in a way that people understand where the direction is, where things are sitting in the grand scheme of finding treatments and cure, which is ultimately the goal of the research that OMF is funding. So we start up with the top left quadrant. We talk about data exploration, which is largely about building information in this field of ME/CFS. This is about comparing ME/CFS people to healthy controls, to other diseases, to determine things that are different, so biological differences between them. And so those biological differences can go to be useful for finding potential biomarkers, which would then move them into the designing of diagnostics. Using these sort of biomarkers to design diagnostics specifically for those biomarkers would then lead to an area where we want to validate them. So at the same time, we're also funding the design, I guess, and build, especially at Stanford, the building and developing of these novel visual diagnostic technologies. And they are really trying to fulfill a need that may not be there, because technology is what kind of holds back progress in most fields in research. And that can definitely be the case for ME/CFS. And so we can see with the new technologies that are being developed at Stanford, you can see ways in which these things can be useful that weren't there before. And so what we'd be hoping to do is validate those diagnostic technologies. On the other end of the spectrum from those biological differences, you can also use that information to generate hypotheses to understand the molecular basis of ME/CFS. And so when you go through that, you then test those hypotheses, you validate those hypotheses, and then you move towards, I guess, developing treatments, which is the end goal. And so as you can see, the real flow from this graph or this outline is just to show that all the arrows are really leading to this area. And so when we talk about a certain type of project that is being funded, we want to keep that in mind, just so that people know where this sort of area and where the progress is heading. And so we're going to start with talking about the research that is being done at Stanford that OMF is funding. So first of all, we have the severely ill patient study. This was obviously the first major study that was funded by OMF through Stanford, through the Ron Davis Lab. And this is a multi-omic deep dive into people with severe ME/CFS. So we're going to use the word multi-omic quite a bit here. Multi-omic is referring to things like proteomics, genomics, metabolomics, transcriptomics. So that's the multiple omics that we're talking about. If you want to break that down even further, something like genomics is looking at all the types of genes that exist within the human body. So all the genetic material, all that information that is passed on through generations will be spread through the multi-omics. Through the genomics. Through proteomics is the same concept, but with proteins. So looking for all the proteins within the body. And then the same concept with metabolomics is looking for all the metabolites. So metabolites and proteins really work together. So genes actually code for proteins, and the proteins will manipulate metabolites into different molecules. And that's really how the basis of all cellular life exists in the body. So by trying to find out all those small little compounds that exist in the body, you can kind of learn how these things are working together. And so when you do these things together, multi-omics, then you get a richer amount of information, because you're not just finding what the little compounds are doing, like the metabolites, you're finding what the proteins that move those metabolites are doing. So that's really the value of multi-omics. So I just thought I'd take a couple minutes to explain that. And so doing that into the severe ME/CFS has been extremely valuable. It's likely that the people within this study had more tests put on them than I think any other patient samples in research history, which is just a testament, I guess, to the complexity of the disease, but also the commitment shown to find something significant within them. And so there is some interesting results coming from that, which we won't really go over today. So the expansion of the Open ME/CFS Data Center to share results as rapidly as possible and build a network for statisticians. So this is an online data center where results can be deposited, speeding up the time in which other people have access to those results globally, people working with Open Medicine Foundation, but also other researchers that just have an interest and want to see what type of data that they're dealing with, see if they can find certain patterns in the data that might be significant of ME/CFS. And it's a great way of building that network of statisticians. So it's almost like, I guess, bait off a boat if you want to go fishing for other scientists that could get into the field. So the Family Big Data Study. So this is also another study that's being funded. The multi-omic, again, characterization of samples from people with ME/CFS and their close non-ME/CFS relatives. So, and this is to find commonalities and differences. So we're dealing with people who are close relatives. So you're looking at for people who have genes that are quite similar, who live probably in a similar environment. And so there will be commonalities between them rather than just comparing them to any group of healthy controls. You want to try and compare them to those who are most closely resemble the people who have ME/CFS and working out what makes them different. Why would ME/CFS be within this person and not this person? So that's really the crux for that sort of work. We're also moving on to the characterization of T-cell signatures in ME/CFS. So this is to determine if there is an autoimmune or an infection-based problem in ME/CFS to do with the immune system. Searching for pathogens in the blood using state-of-the-art genomic technology. So this is looking for common pathogens, ones that we know about, viruses, bacteria, all the ones that we know of, but also the rare pathogens. So these are other types of bacteria, other types of viruses, other type of protozoa, different type of little pathogens that can exist that are rarely ever seen, and also the newly discovered pathogens. So pathogens are changing all the time, and there are new ones popping up, and new ones that we actually are discovering all the time. And so by using sort of newer technologies for looking at the genes and looking for those that aren't human is a way of trying to identify if there are new pathogens jumping up. And so onto the last one is looking for metals in the hair of people with ME/CFS. This is really to look for environmental exposure and the type of compounds that could be existing within people with ME/CFS, especially metals, which can be extremely detrimental and causing a number of the symptoms that people with ME/CFS may have. So most people will probably be familiar with the recent publication from the nanoneedle. So this is a device that uses an electric current and cells pass through that current. It's looking for the way the cell is impeding that current. And so every sample that goes through, the cells themselves have a current passing through from one patient. That normalizes what the current should be expecting to be. Then you add a salt stress and you see how rapidly that current is changing, which is how much is that cell changing in response to that salt stress. And that could be in a number of ways. It could be changing its structure internally, externally. It could be dying. It could be doing a number of different things. And that signature that it creates is significantly different between the ME/CFS patients and the healthy controls that have been run at this point so far. And that's been in 100%, which is obviously very exciting. At this stage, they're testing other diseases, testing more ME/CFS patients, and also testing potential treatments through this device, which is looking if you can ameliorate or if you can add something to those samples so that when the salt stress is added, you don't get that change in signature. And so then number 8 is the developing of a device that measures blood flow and deformability of red blood cells. So there was a paper on this earlier this year. This is about creating a device. Oh, sorry, I'll go back a little bit because I forgot to put up the image for the nanoneedle. So this was the significant variation that we showed. We have obviously the controls here and the ME/CFS patients here. So this was work that Stanford has done on this nanoneedle, and you can see that significant variation between the two, which is very exciting to us. Developing the device that measures blood flow and deformability of red blood cells. This is what the device looks like. You're passing blood through these little channels. These little red blood cells, as an image, flow through. If they're highly deformable, like healthy red blood cells, they can pass through smaller channels. The red blood cells themselves can shrink in size, or can squish down in size and pass through like little bubbles through a straw. If they're not as deformable, then it takes them a lot longer to get through those areas. And so this becomes important when you're talking about peripheral circulation, because all those little channels within your fingers and within the outer areas of your body are very small circulation areas that require red blood cells to be able to be deformable to then bring oxygen to those areas. So this is why this is a significant area of interest for ME/CFS, and they have shown different signatures with this sort of work in ME/CFS patients where their red blood cells look less deformable. So then we have the metabolic trap. So that's work looking at mutations in the IDO2 gene. That came out of the severely ill patient study, which we talked about in number 1. So that has been observed, that this IDO2 gene mutation has been observed in all ME/CFS patients tested so far. That's 66 patients in total. It's not an uncommon variant. It does happen in the large population to a significant degree, but not to the level of 100%. And so the metabolic trap theory is currently being tested in the laboratory, where they're looking at ways to look at the metabolites changing through cells of people with ME/CFS to see if there is a malfunction at this gene functionally, because sometimes you can have mutations in a gene, but doesn't necessarily mean that that's altering the functionality within the cell, because there are other things that can occur. So that theory still needs to be tested and is being done so. The magnetic levitation device is another device. It enables immune cells to be separated by density. So you get a drop of blood like you do with the nanoneedle. What this does, instead of passing blood through a channel, it's actually passing blood through this, magnetic gradient, which allows the cells to be separated into different levels based on how dense they are. So, which is how heavy they are, and in that way, you can sort out whether there are differences between people with ME/CFS and people who are healthy. And so you can get changes in things like immune cells. You can have lower density because of certain diseases. And so this is something that's valuable, worth looking into more to find differences in ME/CFS and healthy. There's also the validation of the ME/CFS metabolomic study, which is being conducted by Dr. Robert Naviau in conjunction with Stanford. So he's at University of California in San Diego. And there's the testing of diagnostic technologies on same samples to determine which is the most effective. So all the diagnostic technologies being looked at, the nanoneedle, the RBC deformability, the levitation, the metabolomics, all these different types of ways of separating ME/CFS and healthy people are all being tested on the same samples to see, one, which is the most effective, but two, if you can use a couple in unison to be even more specific. So rather than just relying on one, you use two, and you can separate out all the patients from the healthy controls. So now we're moving on to the Harvard ME/CFS collaboration. Unlike Stanford, which has been going along for a while, the Harvard collaboration has really been a startup from last year. It's definitely working with groups that are already been working on ME/CFS, and this has been to the advantage of the collaboration. Part of the value of what the ME/CFS collaboration at Harvard can offer is the number of MDs who also have PhDs who are working on research in this field. So it's not just purely lab-benched research. This is finding clinicians who will be doing research as well. And that allows you to broaden the type of sampling that you do and the type of testing you do in real time within the body, which is obviously fantastic for a disease like this. First of all, they want to do a multi-omic deep dive into the structure and function of skeletal muscle in ME/CFS patients. So this is being done at the MGH, which is the Massachusetts General Hospital. They also want to do that deep dive in the structure and function of muscle against mobility matched UK normal volunteers. So what we have here is we have two similar studies going on, one in the US and one in the UK. One that's looking at the functionality based on the technologies they have available at Harvard, and then also replicating a similar type of study at the UK. The interesting thing about the UK study is that it's against these sedentary controls, which are going to be very valuable because there are obviously a lot of things that physiologically can happen from being sedentary for a long period of time or being deconditioned. That in itself does have a significant effect within the biology of the people. So having them as a control is a great way to see if there are the differences. A lot of the differences we're seeing may be related to that deconditioning rather than the disease itself. So we're very excited for that study to be ongoing or beginning at this stage. Both studies are unique at the same time because of the fact that they're looking at skeletal muscle. So the biology of all different areas of the body is different. So if you look in the blood, it's going to be different to actually what's happening within a blood cell that's in the blood, which is going to be different from what's happening in a liver cell, which will be different from what's happening in a muscle cell and a brain cell and a kidney cell. They all have different functionalities, different metabolism, different way in which they function. And so there is likely that these differences we might be missing something by looking at things like the blood only. So having the opportunity to look for a tissue within the body will actually throw up things like accumulations. So things that have accumulated within cells, not things that are running through the kidney and getting removed from the body. So there's a high value to looking at skeletal muscle. In particular, this sort of work is going to be done before and after exercise, which is something of value because a key message that the Harvard or that Dr. Ron Tompkins is trying to get across is that he wants to characterize not just ME/CFS, but post-exertional malaise, which is significant of ME/CFS, and what we see as being the significant symptom of the disorder that separates it from a lot of other diseases. So trying to find the answer or the biological underpinnings of that symptom is looking like the best way to go. And this is why this type of work is being undertaken. So there's also going to be a multi-omic characterization of blood samples before, during, and after ICPET. So ICPET is what they call invasive cardiopulmonary exercise testing, which is when they have someone on a bike riding and they have, this is invasive, so they actually have little measurement or little measuring tools that go up through the veins on either side of the heart, so the arteries and the veins, to measure the functionality of the heart during that test. So it is quite invasive. But by doing that, they can take blood samples from all different areas of the heart before and after it's getting oxygenated. And so they can learn a lot about it. This is a standard type of test that actually does happen for a lot of different conditions. And so they're lucky enough to have clinicians at Harvard who are running these tests, and a lot of them are finding ME/CFS patients going through those, and they're collecting those samples, and then we're going to be doing, or Harvard are going to be doing a multi-omic characterization of those samples. So there's also looking at the role of neuroinflammation in the brain of ME/CFS patients at MGH. So neuroinflammation is obviously a big deal in the field of ME/CFS at the moment. Neuroinflammation, the way that you look at it is using brain imaging, and they have a really great setup at the MGH through the Martino's Center, looking at all different types of brain imaging, different ways they can look at people's brains and determine whether neuroinflammation is having an impact in ME/CFS and whether it's specific for ME/CFS, and if there are ways in which that can be ameliorated or if you can remove that neuroinflammation. Also looking at the role of preload failure. So I mentioned the IC PET at number 3. Well, that work has found results of what we call heart preload failure in a subset of ME/CFS patients. That is where their heart is actually not filling with blood the way it should be. So when you exercise, your heart is pumping, and part of that pump working is that when it relaxes, it's drawing in more blood to then pump back out the other side. But in this stage, what's happening is that when it's relaxing, it's not drawing in the amount of blood it should be. And so it has less blood to pump out. And so that's happening in a number of ME/CFS patients. So they're trying to characterize why that might be occurring and what impact that might be having on the biology of the ME/CFS patient. So these last 3 are very interesting. They're more in line with trying to find things that can impact the community as fast as possible. What we're talking about is the use of computerized adaptive testing, which is looking at taking clinical data or information that are already existing and working out ways in which we can improve diagnosis as it stands by finding different types of tests that might be doing a better job of separating ME/CFS patients from other diseases. So working out which type of symptoms might be doing the best job, whether we can narrow that down to simplify the diagnosis for other clinicians and other GPs worldwide without making it too simple that they misdiagnose the condition. Number 7 is raising the awareness of public education, but also the education within these clinics and the hospitals around the MGH and the Brigham and Women's and the Beth Israel hospitals. So they will be looking at ways to raise awareness and educate the groups around there. And so this is a great opportunity to try and get the name of ME/CFS through the channels of medical education. And so the 8th is obviously setting up the long-term goal, which is to set up the establishment of infrastructure to do clinical trials, to support multi-center trials on ME/CFS through the Harvard Collaborative Initiative. So these 3 are really great at this time point. We're trying to aim to get to these because obviously they can have a significant impact as soon as possible on people with ME/CFS. And so last but not least, we're going to go through the Uppsala ME/CFS collaboration research thrusts. We're starting with sampling and analysis strategies for biosamples. So this is looking at ways in which they sample from ME/CFS patients. The unique thing about the Uppsala collaboration is that they actually get cerebral spinal fluid from ME/CFS patients, which no other clinic or other groups worldwide, as far as I know of, have access to at this stage. And so they're trying to perfect the way to do that with minimal invasiveness. They're also trying to perfect the way of collecting blood samples. So instead of going to a doctor and getting a syringe or a needle in your arm to collect the blood samples, they can collect samples, or they're developing the way to collect samples with a blood prick, just a prick in your finger and just getting a blood spot on a dry piece of paper, and then having that being the way that they collect these samples. So this is important for many different reasons. If you want to take a number of samples from people over a short period of time, they don't want the pain. Obviously, ME/CFS people are susceptible to this type of to constant venipuncture, and also you have the ability to get to people who are at their homes, so severely ill ME/CFS patients. And so then they're looking at the untargeted and targeted proteomics of neuroinflammatory markers. So this is in particular looking at the cerebral spinal fluid and looking for markers or metabolites or proteins or in this case proteins, that may be significant of ME/CFS as compared to other diseases like multiple sclerosis and fibromyalgia, ones that may have some similarities. And just to determine what differences, what makes ME/CFS significantly different from those different diseases. So then we also have autoantibodies and autoimmunity that's being looked into ME/CFS patients. So this is looking for proteins or antibodies within the body, within the blood, which may be targeting the self of the person. So what you have are these antibodies that will look to attach themselves to elements of the person that isn't a pathogen, but actually their own tissue. And so that's what they're looking for, to find if there are any antibodies like that in ME/CFS patients. Targeted metabolomics of dysfunctional steroid and thyroid hormone regulation. So obviously the endocrine system, the hormones in ME/CFS patients, there has been a significant amount of research on this in the past, and that kind of has died down over the last few years. So they're really bringing this back because it does serve a prime function. If you're going to have a multi-system illness, you'd probably suggest that hormones are having some role to play in that, considering that they do create the changes throughout the body that may be reminiscent of a multi-system disorder. So this is interesting to look at these type of hormones specifically. And so they're trying to find differences between ME/CFS patients and healthy controls. They're also looking for trace elements in ME/CFS patients. They're looking for microglia imaging with PET in ME/CFS patients. So again, this is looking for the neuroinflammation. Microglia are the immune cells of the brain, and by imaging them, you're trying to determine whether they are primed or whether they are active against some sort of infection or creating an inflammation event. And so that's what they're looking for as well. You have intranasal mechanical stimulation treatment for ME/CFS. So this is and there's also probiotica treatment and microbiome metabolome in ME/CFS. So these are two treatments that are being studied. And I think in collaboration, the group at Uppsala are going to be looking at the metabolome, looking at the metabolites, the proteins, to see what's changing in ME/CFS patients through these treatments being conducted, to see if they are actually having a positive effect, and what, if they are having that positive effect, what is changing in the biology of those ME/CFS patients. They're also doing an assessment of the immune response, a large-scale study with clinical and laboratory blood tests to see differences between ME/CFS patients and fibromyalgia. Again, the nice thing about this sort of work is that you're no longer just comparing to healthy controls, you're trying to compare to other disease models that look similar to find things that are distinct of ME/CFS. And now studies on newly discovered anti-mitochondrial peptide or new antibodies in ME/CFS as well. So more work on looking for autoantibodies. So again, I just bring back to the basic overview. You can see a lot of the work is looking at building of data, but also designing of diagnostics, designing pathogenesis, and developing treatments. I'm just going to hand it back to you, Linda.
Well, thank you very much, Chris. That was wonderful and a great overview. And as you can all hear, we have a lot going on. There's a lot going on out there, both within our collaborative centers, and also, which is great, with people researchers who've been doing research in this space, in ME/CFS and other chronic complex diseases, that have been doing research that are now going to collaborate with us. And so we are excited about all this. We're excited about all the expertise that's going to be part of this. And we want to thank you again for helping us continue and scale up all these efforts. There's a lot of plans on the table, and there's a lot of things that we would like to do, that we want to really scale up and move this as fast as possible. And we could not do this without your help. So we want to thank you again, and thank you for joining us at our confidential update here. If you have any additional questions, I'd like you to feel free to email us at omf@omf.ngo, and we'd be happy to answer those questions as well.
Ich würde noch gerne was sagen zu Linda Tannenbaum. Diese ganze Forschung, die jetzt hier präsentiert wurde, ist zurückzuführen auf die Initiative von Linda Tannenbaum und dem Professor Ron Davis. Linda Tannenbaum ist die Mutter einer kranken Tochter und Ron Davis Vater eines kranken Sohnes. Und Linda Tannenbaum hat die OMF im Jahr 2012 gegründet. Und Ron Davis hat im Jahr 2014 ein erstklassiges Forscherteam zusammengestellt, das seitdem immer mehr gewachsen ist. Die Forschung bisher wurde im Wesentlichen durch Spenden von Betroffenen und deren Familien finanziert. Und die Ergebnisse daraus haben jetzt dazu geführt, dass auch endlich Gelder in den USA von der öffentlichen Hand in die Forschung dort fließen. Trotzdem ist die OMF nach wie vor auf Spenden angewiesen, und deswegen wollte ich alle Zuschauer, alle Betroffenen bitten, einen kleinen Beitrag dazu zu leisten im Rahmen eurer Möglichkeiten. Jeder Beitrag hilft, und es gibt dazu auch ein Spendenkonto, wo man in Deutschland spenden kann, auch gegen Aushändigung einer Spendenbescheinigung, die man dann auch bei der Steuererklärung angeben kann. Ja, danke. Wir haben hier ein paar Blätter dazu. Wer daran interessiert ist, kann sich dann was holen. Also wohin man spenden kann und wie es abläuft, alles dort beschrieben. Okay, danke.
Wo wir gerade das Thema Spenden angehen, wir können es auch so machen, dass, wenn Sie spenden möchten, auch an den Fatigatio, dass Sie wirklich eine zielgerichtete Spende abgeben. Wenn Sie eine zielgerichtete Spende abgeben für Forschung oder für irgendwelche andere Unterstützung, ergänzen Sie das, und dann fließt das direkt auf das Konto. Und wir können auch dann versuchen, das gezielt an die USA, an die OMF weiterzuleiten. Wir können aber auch versuchen, Stipendien zu installieren, um Forscher in Deutschland zu unterstützen. Das kann man dann alles mitsehen.
Hi Chris. Hi, welcome.
Hello.
Welcome to Stuttgart.
Oh, thank you very much. It's good to be here.
Thank you very much for joining us and help us with our questions. And we have Dr. Ebel, who would like to translate the questions of our auditorium and your answers. Okay,
great.
We have the first question there.
Schuster. Und ich glaube, ich mache es in Deutsch, ich kriege es in Englisch nicht hin. Und zwar kam mal auf Fibromyalgie und CFS. Habe ich das richtig verstanden, dass sich das gegenseitig ausschließt? Weil wir haben in unserer Gruppe das letzte Mal die Frage gestellt, wer ist von Fibromyalgie betroffen? Da waren es, würde ich mal sagen, so 70-75%. Kann man die alle praktisch dann, wird das getrennt oder ergänzt sich das? Und eine zweite kurze Frage noch. Bei mir wurde vor vielleicht 3 Monaten Liquor entnommen. Das hat geheißen, ja, wird ein bisschen mehr entnommen, kann man vielleicht noch was untersuchen. Kann man da eine Liquorspende machen? Also so zum Untersuchen zum Beispiel.
Okay, there are two very different questions. The first is the question to separate or to differentiate between CFS and fibromyalgia. Some kind of your talk suggested that there will be a separation and there will be results of your research to make a better differentiation between these two diseases. Is it the goal of your work to differentiate, or is it something that is happening?
Okay, so we see a separation between fibromyalgia and ME/CFS in the US and in Australia. Their diagnosis is different because of the type of symptom expression that they have. Largely, ME/CFS is characterized by that post-exertional malaise, and fibromyalgia is largely centered upon the tender points and the pain. And so what they are doing is trying to find diseases that have similar symptom sets that are to be used as a control, because what we want to see is, you know, it's one thing to see someone who's healthy versus someone who has a disease, but it's another to see the difference between someone who has a disease that's similar and what makes this disease different from that one is kind of what we want to use those for. And we call that in science a positive control.
Okay, I will give the translation in German to the asking person. Das erste Ergebnis ist, dass ja die Klinik, also die Symptome der beiden Erkrankungen sich unterscheiden, weil die postexertionelle Malaise, die Verschlechterung nach Belastung gibt es ja bei Fibromyalgie in dem Form nicht und die Tender Points gibt es dafür bei CFS nicht so. Und das heißt, es gibt schon Bestrebungen, das voneinander zu unterscheiden und es gibt schon erste vorläufige Ergebnisse, dass im Proteomics-Untersuchungen da Unterschiede sind. Und dann würde sich daraus ja auch ergeben, dass dann wahrscheinlich eine andere Behandlung da ist. Ja, ich denke, darum geht es genau, zu sehen, natürlich kann ein Patient Läuse und Flöhe haben. Ne? Und das heißt, natürlich wird es Patienten geben, die beides haben, aber das würde man mit diesen Untersuchungsmethoden dann ja auch herausfinden können. Jetzt muss ich nochmal fragen, was die zweite Frage war, weil das habe ich jetzt vergessen. Yeah, genau. The second question was the patient here in the auditory asked, she had already given some kind of liquor for an examination and she asked if there is something like a liquor bank or something to give a donation for scientific reasons. Is anything about you are thinking?
So we don't have, we call that a biobank and we don't have one. But there is one at the UK. In the UK, there is a biobank for ME/CFS there. I'm sure that they would be willing. But we do take samples from different groups in different areas, depending what you want to do. But if you want to, you can get in contact with us. I think we have contact information on omf.ngo at our website, and so you can always email and suggest that you'd be interested in doing that, and we can put you on our little registry of sorts.
Okay, perhaps from the medical point of view, it would be easier not to send the liquor from Germany to the US because perhaps the probe is not very suitable when it is coming to you. Okay, thank you. I give it to the asker. Es gibt eine Biobank in England und die sind dankbar für Spenden jeglicher Art. Problem ist natürlich die Haltbarkeit der Spende. Wenn man das hier einfach nur in Briefumschlag tut und nach Amerika schickt oder England schickt, ist die Frage, ob es sinnvoll ist. Er bietet an, dass man sich dort über die Website oder per E-Mail melden kann, wenn man gerne Gewebe spenden möchte, jeglicher Art oder Körperflüssigkeiten, dann kann er entsprechende Kontakte vermitteln. Das muss man dann sehen, ja. Ist sicherlich sehr speziell. Wie gesagt, es ist schon schwierig, innerhalb von Deutschland zum Beispiel mikrobiologische Proben zu versenden oder für bestimmte Blutuntersuchungen, die innerhalb von einer Stunde im Labor sein müssen. Also wenn man jetzt den Liquor irgendwie nach Amerika schickt, ist es vielleicht gar nicht so richtig sinnvoll. Ne?
Hi, thanks a lot for this amazing overview of the exciting science going on. I have a potentially naive question on the red blood cell research you're doing. I was pretty intrigued that some can be soft and like healthy and deform, and others can be pretty hard. Is it known what makes them less deformable? Do they have like different composition of the membrane? Do they have problems in cytoskeleton turnover? And is it like a cause or a consequence due to metabolic defects in patients?
Yes, that's a good question, and that's something that we are trying to look into more as well. But definitely, the cytoskeleton defects is a key component. Specifically, oxidative stress is something that is altering the composition of the red blood cells so that they aren't or are less deformable. But there's some interesting studies into it, and this is kind of an area that is growing not just for ME/CFS but for other diseases, because it is something that's important for other diseases as well. But the dynamics of the red blood cells are very intriguing. Just when they're deformable, they can slip through capillaries much easier. But some of the things that we really care about is also not just whether they can get places, but whether they can give oxygen to the tissues. Because being able to squish down and travel through small capillaries actually increases the surface area of the red blood cell to the tissues, enabling more oxygen transfer. It's an important part of getting oxygen into cells. So that's a key element to it, and from what we've seen, the less deformable still have an ability to do that. It's just a little bit altered. But in terms of what's causing those problems, we're thinking oxidative stress is one element. There could be metabolic problems internally. There could be just a general predisposition to have an issue. And yes, as always, collagen and cytoskeleton is a big component of cellular structure.
Okay, die Frage war zur Verformbarkeit von roten Blutkörperchen. Das hat man ja auf der einen Grafik gesehen, dass die sich nicht mehr so in die ganz engen Röhren, also nicht mehr in die kapillaren, ganz feinen Blutgefäße hineinbewegen können. Die Frage war, ob es jetzt schon bekannte Faktoren gibt, woran das liegt. Die Antwort war, dass verschiedene Sachen dazu beitragen können, zum Beispiel Stoffwechselprodukte oder auch allgemeine Entzündungsstoffe, die dazu führen, dass die roten Blutkörperchen sich dort nicht mehr so verformen können. Aber es ist eben sehr, sehr wichtig, auf diese roten Blutkörperchen zu schauen, weil sie ja den Sauerstoff ins Gewebe bringen. Und da, wo sie nicht hinkommen, da können sie keinen Sauerstoff hinbringen. Und das führt natürlich zu einer schlechten Versorgung des Gewebes und damit natürlich auch, sagen wir, zu Sauerstoffmangel, Stoffwechsel. Und da sind wir wieder bei den Stoffwechselwegen, die wir schon mal gesehen haben, Laktatproduktion und schlechte Energiegewinnung. I would like to address you myself. I'm a doctor for intensive care medicine, and when you describe this phenomenon of red blood cells, I remembered all these things that are being done in the sepsis research, because the problem of microcirculation and activation of blood cells and this phenomenon of I'm missing the word, bad perfused areas in the body, the same, I think. Did you talk to sepsis researchers?
Yes, we did. This sort of technology actually was being developed at Stanford for a number of different diseases, not just ME/CFS. But we looked into it specifically because of the interest in the fact that we were seeing a difference. And so, yes, this technology was developed for sepsis, for a number of other diseases. This is something that occurs from chronic stress conditions, but it's trying to work out whether there are, not just that it's happening, but whether there are signatures that are specific of different diseases in the way that it's occurring.
Okay, thank you. Ich habe selbst die Frage gestellt, weil ich ja mich auch sehr intensiv mit Intensivmedizin beschäftigt habe, als ich noch arbeiten konnte. Die CFS-Forschung beschäftigt sich auch sehr mit Durchblutungsphänomenen, weil im septischen Schock es auch dazu kommt, dass die roten Blutkörperchen sich eben nicht mehr so verformen können, nicht in die Peripherie, in die Gewebe hinein können, das Gewebe schlecht durchblutet wird. Da spielen auch noch Gefäßeffekte eine Rolle und ich habe ihn gefragt, ob er auch sich mit Sepsisforschern verständigt über die Mechanismen, die bei der Sepsis zu der Veränderung der roten Blutkörperchen führen und da sind durchaus Schnittstellen. Dieses Untersuchungsinstrument mit diesem Röhren, was er uns gezeigt hat, das wird für viele Krankheitsbilder eingesetzt, also für CFS, aber auch für Sepsis zum Beispiel.
Question. Hi, Dr. Armstrong. Thank you for being here. I take this opportunity. I'm Dr. Brambilla from Italy. I had this thing in mind for some time and I never had a chance to ask you or anyone who does research. Is there any ongoing research or study about relationship between malaria and CFS? I'm thinking of different mobility of red cells and Plasmodium is one of the parasites that affects red cells, and there are some symptoms which are common like fatigue and chronic malaria, and I don't know, is there anything going on about this or any idea, any suggestion that developed in any study? And sorry, last thing, I never heard about CFS in Africa. What do you know? What data do we have about it?
Okay, that's a good question. So no one is doing a combination of malaria and ME/CFS research that I know of. Maybe someone is right now and hasn't publicly made that aware, made anyone aware, but that is something that would be pretty interesting. And in terms of Africa, I know that there is some group doing some work in South Africa, but that's about it, as far as I know. Yeah.
Okay, die erste Frage war, ob es eine Verbindung zwischen Malaria und CFS gibt, weil bei Malaria es auch dazu kommt, dass rote Blutzellen nicht mehr so verformbar sind und auch viele Symptome ähnlich sein können wie Entzündungssymptome und Schwächegefühl und Fatigue. Und die Antwort hierzu war, dass es dazu derzeit keine bekannten Forschungsergebnisse gibt, aber dass es natürlich sein kann, dass jemand daran arbeitet und es noch nicht publiziert hat. Und die zweite Frage war zum Thema, ob es irgendwelche Berichte aus Afrika gibt über CFS. Und hierzu war die Antwort, dass es aus Südafrika, was ja eher dann eine etwas höher entwickelte Gegend vielleicht ist, Forschungsaktivitäten wo gibt, aber dass da jetzt noch keine großen Neuigkeiten gibt, die über das hinausgehen, was wir bisher gehört haben.
Also zu den Forschungsaktivitäten in Südafrika weiß ich, dass Herr Professor Sadra Shirazi Stark momentan eine Ärztin in Hamburg hat mit einem Forschungsprojekt, aber was das jetzt ganz genau beinhaltet, kann ich nicht sagen. Also ich weiß, bestehen von ihm aus Kontakte nach Südafrika, man kann das natürlich nochmal mit nachvollziehen. Vielleicht magst du es.
Ja, wie sieht es mit weiteren Fragen aus? Gibt es noch etwas?
Eine Frage, es gibt in Deutschland und Europa die physikalische Gefäßtherapie BEMER, die ja die Mikrozirkulation verbessert. Gibt es da auch schon Erfahrungen bei CFS und eben auch in USA Forschungsergebnisse?
Das sagt mir jetzt gar nichts. Was ist denn das? Weil wenn ich es übersetzen muss, dann muss ich wissen, was das ist.
Da wird ein Signal übermittelt an den Körper über, der Träger ist ein Magnetfeld, aber das Signal selber gibt praktisch den Zellen der ganz kleinen Kapillaren zu den nächst größeren wieder das Signal, dass die zusammenarbeiten und dass dann eben auch in Filmen darstellbar die Mikrozirkulation sich deutlich verbessert.
Okay, wie genau wird das geschrieben?
Physikalische Gefäßtherapie BEMER, B-E-M-E-R. BEMER,
okay. Bitte? Okay. Here's a question about some kind of physical therapy using a magnetic field called BEMER therapy, a magnetic field to improve the microcirculation. Do you know anything about it, or are there any results?
No, I actually don't know anything about that, but I can look into that. BMER is that what it is? BMER?
BEMER.
Okay, I can look into
this
that.
is a company what is distributing a device to use it some kind of magnetic field application. BEMER. This is the company.
Yeah, that's something that I haven't heard of, but that's something I will look into. Thank you.
Okay, thank you. Also er hat bisher keine Erfahrung, aber er schaut mal nach und überlegt sich dann was. Ja.
Hi, my name is Matthias Bietke. I'm a machine learning scientist, and I was wondering, so I just registered for your ME-CFS data center. I was wondering how many people are already working on your data.
I think there's about 20 at this stage, or just under. But there is a lot of data there, and it's I guess we don't know who's working on it. We just know who's got access to it at this stage, and so we'll find out, I guess, as more papers and more analysis comes out, what's really happening from that.
So I just saw it today. So how long are you recruiting scientists already there?
Yeah, so if you apply for it, it'll usually take a couple of week turnaround. I think it's relying upon a scientist there who works. He works both at Harvard and Stanford. He's a very busy guy, but he will give you access eventually when he gets around to it. It should be within a couple of week period.
Right, thanks.
Okay,
No worries. Thank you.
Die Frage ging um den Zugang von Wissenschaftlern zu den Daten, die von der Open Medical Foundation bereitgestellt werden aus diesen ganzen vielen Forschungsprojekten. Da kann man ja als Forscher sich registrieren und dann auch mit den publizierten Ergebnissen dann weiterarbeiten. Und das war jetzt eine spezielle Frage, wie man dort rankommt. Ich denke, fürs allgemeine Publikum. Also man muss sich dann an ihn wenden und sich registrieren lassen und kann man als registrierter Forscher dann mit den Daten arbeiten. Ja, ich würde auch sagen, letzte Frage, dann
okay.
Ist es lang.
Yeah, hello, I have a question. Is there a connection between CFS and mast cell activation for a subgroup of patients, or are these separated diseases?
That's a very good question because that's something that we are looking into to determine whether that is something that it seems to be something that's quite common in ME-CFS patients. It seems to be something that is the ME-CFS patients themselves will look to treat with antihistamines and work like that. So, and there has been some gene modules that are associated with ME-CFS. One called, I think, the RCCX module, if you want to look that up, which is a gene module that ties together mast cell activation, postural orthostatic hypertension, and then also I think the third one is collagen problems, or EDS. And so that's the kind of trifecta that seems to be somewhat common in ME-CFS patients. So yes, there is some work looking into that connection.
Okay, thank you. When we heard these genes you named, we couldn't really be so fast to learn that. Could you please mail that to Fatigatio? Then
yeah, sure.
That could be very interesting. Thank you. Die Frage war, ob es eine Verbindung gibt zwischen CFS und Mastzellaktivierung, also auch Histaminausschüttung. Und die Antwort war ja. Sie haben ja gesehen, er hat gelacht. Das ist offensichtlich also schon ein Hot Topic, wie man so schön sagt, also ein heißes Thema. Die Befunde, die es bisher gibt, zeigen, dass es da offensichtlich eine Verbindung gibt und dass auch das Histamin eine Rolle spielt. Er hat verschiedene Gene genannt, die man schon nachweisen konnte in dem Zusammenhang. Das müssen wir dann nochmal mailen lassen, weil das war jetzt nicht zu verstehen. Aber da sind sie sehr nah dran und genau das wird eben zum Beispiel mit solchen Proteomik-Analysen dann auch nachgewiesen wahrscheinlich. Ja, vielen Dank, dass ich helfen durfte.
Erst einmal, ich spreche in Deutsch, das fällt mir doch leichter. Ich hoffe, bitte das zu entschuldigen. Erst einmal ganz, ganz herzlichen Dank für die Unterstützung, für die Beantwortung der Fragen und ja, für das gezeigt wurde, dass Forschung möglich ist und wie Forschung in diesem Rahmen möglich ist und dass wir Einblick erhalten durften, wie das in den USA wirklich von Grund auf geplant ist. Und ich bin sicher, die Welt ist nicht nur eine Kugel, sondern wir können uns um diese Kugel alle die Hand reichen wie ein Netz. Und es ist schön zu wissen, dass wir alle zusammenarbeiten können. Und ich würde mich freuen, wenn wir alle weiterhin in Kontakt bleiben. Vielen, vielen Dank, Chris.
This were the thanks and the greetings from Birgit Goske, and she wanted to thank you for this very great talk and the insights you gave us, and she's very happy that the world is not a flat screen but a sphere, and that we can put our arms around the sphere and go hand in hand to reach significance, better lifestyle and health for the patient, and we are very thankful for your activity. I think it's wonderful. I did never imagine that something like that in this huge amount would be possible. Thank you very much.
Well, thank you very much for the kind words. Yes, we're very, very happy with the amount of research that's happening worldwide. It's tremendous, and I think together we can make a big difference and start a movement in this research area.
Thank you. Thank you very much.
Thank you.
Also er hat auch nochmal sich herzlich bedankt für unsere Aktivität und dass man gemeinsam eben viel mehr erreichen kann als allein.